Welcome to CSEC Biology TCP. Today we're going to be looking at the Human and Social Biology paper for January 2021. It is a paper 2 that we're looking at. We did publish question number 1, 2, and 3 prior to this publication. You'll find the link for that in the description below. So we're going to just start this paper looking at question 4, 5, and 6. Remember to like, share, and subscribe if you like the work that we do. That will enable us to continue doing the good work. Thank you much. Let's go. In our last lesson, we left off here. This is a final part of question number 3. We are going to be moving down now to question number 4 to start this publication. Alright, great. Here we go. Alright, Moist class is asked to prepare a poster which highlights important facts about acquired immunodeficiency syndrome, AIDS. Now we're tasked to name the causative agent of AIDS. Now there are 5 things that really could cause a disease. There could be a virus, a bacteria, fungi, protozoa, and worm. Which is the causative agent here? I'll reveal. It's going to be the virus. The particular virus is the human immunodeficiency virus. And we move on to question 2, which says state one method that can be used to limit the spread of AIDS. One method that can be used to limit the spread of AIDS. Let's look at that. We could use a condom. That could be used. A contraceptive method that could be used is, of course, a condom. And of course, abstinence is always something to have a discussion about. Now, the other question, pregnancies in which mother have contracted a sexually transmitted infection, STI, can result in birth defects. We are tasked, identify 2 STIs which could result in birth defects. Now we know a lot of STIs. We know gonorrhea, we know AIDS, we know syphilis, and the list goes on and on, herpes, and a whole lot. Alright, so let us look at what my answer is here, which of course might not be yours, but make sure that you can substantiate your answer. So let us go. I'm going to be looking at mine. So the 2 diseases I'm looking at here is, of course, syphilis and gonorrhea. Syphilis and gonorrhea. So I'm identifying 2 STI which could result in birth defects, syphilis and gonorrhea. Now the other question states 2 effects that STIs may have on a newborn baby. So 2 effects that STI will have on a newborn baby. Let's look at that. What is it that you have there? I'm going to reveal. So syphilis will of course cause a enlarged liver, and the ripple effect from that is so great it could of course take the life of the newborn. Gonorrhea could cause blindness in the newborn, so one wants to ensure that they are taking all the precautionary measures to ensure that they are safe, and if they are not safe, that treatment is sought early. It means that if one is pregnant, it's very important to ensure that they are going to those maternity clinics. Alright, so we move right along with the other question, and here we have C to this question. Table 1 shows a number of birth per 1,000 in 3 countries related to their different birth control method. Now look at the table here and the construct. That is very, very important, and how the title is given to the table. So for country X, we have condom, which is of course number of birth per 1,045. Now country Y, rhythm method, 65. Country Z, withdrawal of a penis, 70. Now we're tasked, identify the country with the highest number of birth per 1,000 and state one likely reason why the associated birth control method resulted in the highest birth rate. What do you think the answer is here? Let's look at it. So withdrawal, of course, had the highest birth rate with 70, and this is not the most effective method, as there could be early leakage of the sperms, which of course would result in pregnancy, or could result in pregnancy. Hence withdrawal is our answer there for I. Let's look at the other question. The other question, identify the birth control method in table 1 which is most effective against preventing pregnancy. State one reason why this method is the most effective at preventing pregnancy. Let's look at our answer here. Condom is that birth control method. Now the condom forms a barrier that prevents sperms from reaching the egg. Of course, if the condom is preventing the sperm from reaching the egg, then here it is definitely going to be the most effective, because pregnancy will not occur providing that the condom is properly used. Let's look at the other question. It says that we're supposed to suggest two reasons why the number of births per 1,000 when using condoms in country X is still relatively high when compared with the use of the rhythm method in country Y. Now let us look at what our answer is here. Condoms were not properly used. So we're suggesting that the condoms were not properly used, and as a result the number is as high as it is. It could be that it is not properly used, in one sense that it is not properly worn. There are some persons who really don't put on the condom properly, some persons who are using lubricants that are not needed, some persons who are not wearing the right size condom. All those things could contribute to a pregnancy. There are persons who are even reusing condoms. So all this could of course lead to the high number. Condom might have torn, and there was of course leakage of a sperm. So a person might be using the condom, and just before there is ejaculation the condom could have torn, but there might have been early leakage of sperms, and that of course led to pregnancy. So these two methods, or these two things, could of course cause the number to reflect as it does in the table. Apart from having lower birth rate, state one other benefit associated with the use of condom during sexual intercourse. Now this one is going to be very, very easy for a lot of persons. Let me see how well you did as I unveil. So it guards against sexually transmitted infection. By this, most persons would have known that the condom actually guards against sexual transmitted disease, and of course prevents pregnancy. So just two other non-surgical methods of birth control that would be most effective for reducing the incidence of pregnancy. All right, non-surgical. We know that surgical would be like tubal ligation and vasectomy. So the diaphragm with spermicidal gel is of course non-surgical, and of course contraceptive pills, those would also be non-surgical. So that's our answer there. Of course you might have something that is different. Now let us move right along. Question number 5. Explain what is meant by a balanced diet. Now this is going to be a fancy one for most persons. It is a topic that most persons would have on the tip of their tongue. A balanced diet is a diet that contains food from all the food groups in the right amount and provides, of course, adequate nutrient for good health. You will agree that if the diet is not balanced, then we could be suffering from what is referred to as deficiency disease. And in some cases, these deficiency will spill off into other disease. If we were supposed to look at vitamin A and the lack thereof, we could be looking at a weakened immune system, and of course we wouldn't want that. So a balanced diet is definitely important for all individuals. Identify the Caribbean food group that should be consumed in largest amount and the Caribbean food group that should be consumed in the smallest amount. Now here we're talking about food group, not about nutrient. Alright, so please don't think about carbohydrate, fats and protein and minerals, and that's not what we're talking about here. We're talking about food group. So the largest food group is of course going to be your staples. I'm sure that somebody said that. Staples. And the smallest is of course going to be your fats and oil. And of course you can relate to the food group or the food that are associated with staples and of course fats and oil. Remember, as you do your past paper, you work them with your textbook to ensure that you understand the topics well and you are certain that what is presented is what you should of course be getting. So we'll look at B, which state 2 benefits of including fiber in the diet. This is of course another easy one from a lot of persons. They are going to be saying this one right off the tongue. Dietary fiber stimulates peristalsis, hence allowing food to move through the gut easily. Fiber also helps to prevent constipation. Now when we look at this, peristalsis is that movement of the food through the alimentary canal. It requires pretty much a contraction of those circular muscles, squeezing the food through the gut, one would say. And constipation is pretty much when there is a difficulty passing your stool. So of course roughage or fiber is very, very important in the diet. And we look at C. In some developing countries, night blindness is mainly caused by the deficiency of vitamin A in the diet. Alright, and we're talking about developing countries, not developed country. This usually occurs as these countries do not have access to fresh fruits and vegetables that contains vitamin A. We're tasked. Explain how genetic engineering could be used to produce vitamin A. Your response should include the following terms. Plasmid, interesting. A plant cell with vitamin A genes, and of course DNA and bacteria. Once we see this is one of the topics that you have to study for your exam, the genetic engineering recombinant technology, like there is for the water cycle and sewage treatment. If you are doing human and social biology exam, there's no way you can go to the exam without knowing these two topics. They are always littering the multiple choice paper and of course the paper 2. Now this is pretty much a long process, so I want you to look at this, and I'll also show you in this example what would have happened if we were looking at the production of insulin. Let me unveil and take this to you, as by now you should be aware of golden rice, and this is pretty much the same type of technology we're looking at with this particular question. Once you would have seen this question with vitamin A, immediately pop into the brain should be the whole golden rice production. Let me share with you what we have. Now the recombinant DNA technology could be used to provide the vitamin A. Now, how does this work? Now, the bacterium contains plasmid, ring of DNA, that facilitates replication of the bacteria. Now, what we're going to do are of the bacterium. For this process, plasmid DNA from bacteria is removed, then it is cut by an enzyme. One could say a restrictive enzyme or a restriction enzyme, but it is cut by an enzyme, right? Now, remember, enzymes are chemical. A vitamin A gene is cut from the plant cell with vitamin A genes in the DNA. Now, an example of a plant cell that would have this vitamin A is the zea mays, or one might call it corn. Now, the vitamin A genes is then inserted into the plasmid DNA, forming a recombinant DNA. Now, look what happens next. Now, the recombinant DNA is then introduced into, now where I have this blue bacteria, this is what would have happened for insulin production. Ignore it for this question. I'm just using it to teach. So what would have happened here is that the recombinant DNA is then introduced into the desired plant. For example, this would have been the rice, the desired plant embryo. Now, the plant incorporates this new gene into its DNA and produces seeds which will pass the genetic information to produce provitamin. Now, provitamin can also be looked at as, I think, carotenoid, that thing from the yellow part of the carrot, the corn and that type of a thing. Now, what provitamin A does, now the body will change, the body will change its provitamin A that is created as a result of this technology. Your body will now transform that into the actual vitamin A that is, of course, going to be guarding against night blindness and, of course, safeguarding the immune system. So this process is, of course, used in golden rice to produce that vitamin A. So remember, what is produced in the golden rice is the provitamin A. All right, I use provitamin A because it's easier to remember. And then the body will, of course, transform that into actual vitamin A. So remember, when we're looking at this particular question, we are looking at the recombinant technology. It's very important for you to know. Remember, you are going to ignore the bacteria here, because being inserted in the embryo is going to be that recombinant DNA, which is a combination of the bacterium and, of course, that gene from the plant with the vitamin A. So both are going to be inserted into the embryo in order to produce this golden rice. The bacterium there is, as it were, if we were looking at insulin production. Just wanted to drag your memory to the insulin production for those persons who are aware of that. But remember, this technology is pretty much looking at golden rice. There's always an ethical concern around genetic engineering, be it good or bad, and usually a topic that most teachers would ask students to debate. But remember that we're looking at the 2021 paper 2, human and social biology. We would have published 3 questions before. You are going to find the link in the description below. So just go on down to the description and you could watch the first 3 questions first or last, whichever you choose to do, so it will be there for your perusal. Let's look at this question. State 2 advantages of using genetic engineering to produce vitamin A. Vitamin A, we spoke about golden rice. Now, one of the things in producing this vitamin A, as it were, for golden rice, as you would have seen in as precursor to the question, that it is in area where there is a lack of fruits and vegetables. So there is a limited supply of food content with vitamin A. So to use genetic engineering, it would provide a fast fix to the problem of night blindness due to lack of vitamin A in these areas. So this is a very quick fix, right? You don't have to wait a long time. You could just curtail this problem now using genetic engineering. Another thing is that it will reduce the death among children in developing countries as a result of a compromised immune system. So because they really don't have enough vitamin A, or they don't have vitamin A to the diet, we are going to be suffering from night blindness, or they're going to be suffering from night blindness, and they'll have a possibly compromised immune system as a result of that lack in vitamin A. So these are two reasons why genetic engineering is, of course, a great advantage in producing vitamin A in these areas. Now let's look at the other questions and see what they are saying. I do hope that you are following and you are understanding pretty much what we are doing. So here we're looking at another question, and let's read. This is a final question for this particular question. I think we're at question number 5. So there's 2 ways in which genetic engineering could be used to improve food production. I remember when I was going to school and I was doing agriculture, I was told about the maypan, and the maypan is very short and it's easier for the farmer to access the fruit, and also it is a plant that will, of course, produce more food, and that was definitely a product of genetic engineering, and it is with me until today. So genetic engineering shortens the time taken for food products to mature and be ready for consumption, thus improving food security. You would recall days of time ago, we would have to run down a chicken to get a chicken to eat. If you think about the primitive people and so on, the Stone Age guys, now we have birds that are grown in poultry houses and, of course, genetically modified, and within 6 weeks we have chicken to eat, like any common fowl that you would have running around your home here in Jamaica. So of course you'd see that genetic engineering allow you to grow these food material, or these food stock, be it plant or animal, and of course producing food in a very short time. Another important thing here is that genetic engineering also increase yield, and yield refers to the amount that is produced. And we'll talk about the amount that is produced. One might say amount refers to quantity, so we could look at numbers, but we could go a little more than numbers. We could look at the size of the breadfruit, for those persons who know, are the size of the apple, being that apple is a common fruit. We could look at the sugar content as the yield. Is this a sweet apple? Is this something that the market desire? We could look at pumpkin. Is it the right color that the market is looking at? Is it yellow or orange? So here we're talking about genetic engineering and yield. So we could look at the size of a colorless leaf. We could look at the sugar content in the cane. We could look at the sugar content in the apple. We could look at the amount of ackee or guinep, whichever one. I don't know, Barbados use a different common name for, I think, ackee and guinep. So whatever fruit, we could look at the number that is produced per plant, and that of course will all accounts to yield. We could look at how lean the animal is, and we could look at the milk production, right? And we could even look at the speed of the horse, right? Or the size of the bull, or the number of offspring to a particular animal. All those are yield, forms of yield. So yield doesn't really just refer to amount. All those examples would fall nicely under yield. So we say that genetic engineering also increases yield, and thus increasing the world food supply. More is there for the market, and of course it reduces the shortage of food. You would have heard about persons suffering from starvation and persons fighting for food. Even in the Caribbean, we've heard about that. We want to ensure that some students become so skilled at genetic engineering that they will better help the Caribbean, so we can feed all, and there will definitely be less riot and death. Question number 6, which is the final question. Describe 2 ways in which water can be purified at home. Students, please be reminded, do not go to the exam without knowing about water purification, water treatment, and of course about genetic engineering. You really don't want to do that. You don't want to go to the exam not knowing about natural selection and artificial selection. You must know these if you are doing HSP. Please guys, please be reminded you need to know these topics. These are going to be some of the topics that are going to be on my prediction as soon as I get that information from CSEC come May 5th as to what the broad topics will be. I think sometime the 10th of May some student told me. Alright, so we're looking here. We want to describe two ways in which water can be purified in home. In some cases my answers are really limited. In some cases they are a little more, depending on how much I think you know the content. So we could purify water at home by boiling the water. Now when we talk about boiling the water, we're pretty much talking about the water coming to a bubble. All right, bubble, right? So you want that to happen for about 10 to 20 minutes, and that would of course be effective, and of course an effective way of purifying water. Now, like I said earlier, the water must boil. So you're going to get this water. Of course, if it has sediments and of course suspended particles, you want to filter that first. All right, you're going to filter that to remove the sediment, and then you are going to boil this water for 10 to 20 minutes. Now, another method that you could use if you have access to chlorine, it is of course chlorination, where water can also be purified by chlorination. This is of course the addition of a 500 milligram of chlorine to 20 liters of water, or you could use 1 teaspoon of bleach to 1 liter of water. And these are two simple ways of purifying water at home. So if you were supposed to have a hurricane, God forbid, or some reason that you have to get water from a supply that you really don't trust that much, ensure that you do the limited thing that you can do at home to of course purify the water. Let's look at our second question, and this is a lab, and I'm going to break it down a little bit for you as we go through. Now, a few months after an earthquake, 6 students brought samples of water from their respective village to the school laboratory to test for the presence of bacteria. Now, figure 10 shows the agar plates at the end of the experiment. So we're seeing A, B, C, D, E, and F. Now, one of the things we know about this particular experiment is that these white dots would have shown the colony. So the black area on the plate there would have shown pretty much the agar. Now, what we want to look at here, most students, including myself, might be a little affected by the A, right, which seems to be a very poor picture. But we consider that it is not a poor picture, though this is a paper that would have been photocopied millions of times. But providing that it is not a poor picture and it is what we see, we think that it would have more areas of white, which would have represented the colonies of bacteria. So it would have more, followed by B, followed by C, followed by D, followed by E, followed by F. So of course F would of course have the least, while we think that A would have had the most, being that we're just having few little areas of black here, and all this area here would be colony of bacteria, being that bacteria represent the white portion of the agar plate. Let us look at this. Identify the sample which shows the least number and greatest number of bacterial colonies. Now let us look at this. I did say it earlier, so A is going to be my answer for the largest. So largest number, okay, I have something mixed up all here. So the largest number is going to be largest number. Just let me just change that here. Largest is going to be A. Oh, the least. Oh, I had it right. I had it right. So the least number is of course going to be F. Let's look at F. So we only have 2 colonies here. So the least number F has the least, and the greatest number A has the greatest. Remember, we said that A has more white area than any other of the plates, so we consider A to have the largest. Explain 2 ways in which the water sample with the greatest number of bacterial colonies could have become contaminated. All right, so what could have caused this water contamination? Bear in mind, these samples were taken right after an earthquake. So you know what earthquake would have done, would have shaken buildings to the ground, would have, of course, damage and disrupt infrastructure, that type of a thing, and it would not be lenient on our sewage system or our septic pit. So these are some things we need to take in consideration. So we think that the earthquake could have caused sewage pipes to break, which of course lead to sewage flowing into water supply, causing the contamination. We also think that leakage of septic pit, or some might say septic tank, into water tables or other water bodies could also be responsible, as the earthquake could have caused damage to the septic pit or tank. So that moving of the earth crust and that shaking could have damaged the infrastructure and would have caused a leak, and of course the bacterial accumulation in, of course, A, to have had this plate with more bacterial colonies. So we move on to our next question, which is going to be discuss the breakdown of wastewater facility. Discuss how the breakdown of wastewater facility could pose major health problems for members of a community. Now let us look at the word community, because for me it was very, very important to look at the word community. Now in science, community could mean many population interacting. So the birds interacting with the fish and the fish interacting with man and so on. Alright, so that could have been a community. A community could also be in social study where a group of people are settled. So we could look at both, but here I am using the whole scientific aspect of biology to treat with community. So here the sewage facility is very important in managing the environmental impact that could be caused by the release of raw sewage in the environment. Here the sewage is treated, and then less harmful portion is released into the environment. Now, when there is a breakdown in the facility, raw sewage may enter the ecosystem, causing disease like typhoid. If it enters the water body, it will, of course, reduce the dissolved oxygen. So if it enters other water body, typhoid is of course in the water, but if it enters water body, another thing that could have caused is, of course, the oxygen in the water will be used up. That's the dissolved oxygen. It will be used for the decomposition of the sewage that enters the water. Now this is of course going to lead to a death of the aquatic organism, as there's the nutrient content in the water will increase, which will of course lead to eutrophication, and having led to eutrophication, this is of course going to upset the entire ecosystem. The entire food chain will be affected. Now if the entire food chain is affected, then naturally the health of all individuals in the community will of course be compromised. So these are some very important or salient points why we need to have working sewage facility. Here we go. State two methods used in treatment of sewage. So two methods used in the treatment of sewage. This is going to be easy. It is always on your multiple choice paper. It is going to be activated sludge method and bacterial filter method. Now this brings us pretty much to the end of this paper, but there are some things that you need to do to ensure that you are seeing the paper in a timely manner with the most accurate information. Now, to help me produce more of the content you like, it is very important that you do this. You must like the video, right? And then it will go to your playlist. You need to share the video that more persons can be a part of this content. You need to subscribe so that as soon as there's a new publication, it comes to you. When you subscribe, hit that notification bell and select all so you'll be informed. You need to leave a comment and say something about the video. Was it good? Was it bad? Was there something that you preferred we would have done differently, so that we can continue to make great or better video. Now, this will allow YouTube to promote my videos more, which of course will lead to more good videos for you. Please be reminded that the link to the first part of this paper is in the description. Enjoy yourself and look out for other publication. Look to the top of the screen. You can click the next suggested video, or at the end of this lesson you'll see 4 videos on the screen. Please choose one that will help you to grow. Take care.